Yeah, we are live now. Let's start with the formal introduction. Yeah, a very good day to all. A warm welcome to each and everyone present for our SSC E-Talk series, conducted by Saveetha School of Engineering, Saveetha Institute of Medical and Technical Sciences. This is the 13th SSC E-Talk of the series. We are very happy to inform that many have joined through CMAT's Facebook page. I am Dr. Deepak Arun, Associate Dean, International Affairs, Saveetha School of Engineering, Saveetha Institute of Medical and Technical Sciences. At the outset, I extend my special thanks to our Honorable Chancellor, Sir. I also extend my sincere thanks to our Director, Madam, for Madam continuous support, guidance, and motivation to conduct this event in successful manner. I also extend my thanks to Principal, Sir, and Vice Principal, Sir, for their support and motivation. Today we have with us a couple of eminent professors, Dr. Mathew Upton and Dr. Philip Warburton from University of Plymouth. University of Plymouth and Saveetha School of Engineering, CMATs, have signed an MOU and currently working on a lot of collaborative projects. Many students and faculty from Saveetha School of Engineering have visited University of Plymouth as a part of exchange program. Let me take the opportunity to introduce Dr. Mathew Upton and Dr. Philip. Dr. Mathew Upton is the Associate Head of the School of Biomedical Sciences. His area of expertise includes microbiology, molecular biology, disinfection, aquatic biofilms, novel antimicrobials, antibiotic resistance, bacteria, and pathogens. Following PhD studies in Newcastle, he completed a number of postdoc projects in Aberdeen, Antarctica, Bristol, Liverpool, and New Zealand. He is editor of various journals like Journal of Applied Microbiology and Letters in Applied Microbiology. He has published many research articles. He has supervised 20 PhD students and over 70 MSc research projects. He has also received several research fundings. Dr. Philip, he is the lecturer in medical microbiology, School of Biomedical Sciences, University of Plymouth. He holds various professional memberships such as Fellow of Higher Education Academy, Member of Microbiology Society, Member of British Society for Antimicrobial Chemotherapy, and Society for Applied Microbiology. His teaching interests include clinical and environmental microbiology, molecular genetics, and infectious diseases. He has published several articles and received several grants. He also acts as manuscript reviewer for several journals. Now I request Dr. Philip to address the gathering. Over to you, Dr. Philip.
Thank you very much, Dr. Deepak, and thank you for inviting us to speak here today. I can't see anybody because I'm sharing my screen at the moment, so I will go through, and then if there's any questions, I'll invite them at the end, if that is okay.
Yeah, absolutely fine. Please go ahead. Yeah.
Okay. Thank you very much. So today, I'll give a brief overview of the MSc Biomedical Science program that I lead at the University of Plymouth. This is a one-year full-time master's that has 3 specialists. We have a cancer biology pathway, a clinical neuroscience pathway, and an infection and immunity pathway. And it's this pathway that I'm going to focus on for some of the later slides, as that relates to my specialism and that of what Dr. Matt is talking about today. I'd like to point out that our MSc is accredited by the Institute of Biomedical Science, so it is a program that has been seen as recognised to reaching a standard within the UK accreditation service of the Institute for Biomedical Science, and that allows students to then go on and work further in places like the NHS for some of the students that come onto this course. But I will go on to some of the other potential career areas later on. So the MSc Biomedical Science, all students are enrolled onto 3 core modules, which I'll go into detail in the next slide. And then each particular pathway has 2 core modules related to that one. And I've coloured them here in different colours because that will relate to the next slide. So students undertaking a pathway-specific modules related to cancer biology would undertake also a pathway-related project, and that would allow them to graduate with the MSc Biomedical Science (cancer biology). So 3 core modules, cancer biology modules, and a cancer biology project. Similarly, students taking the neuroscience modules, the neuroscience project, would graduate with neuroscience, and the same thing for the infection immunity. We do have occasionally students who want to do something slightly different. So students can enroll onto the course and take the 3 same core modules, take a particular pathway, for example, the cancer biology, and then choose to do an unrelated project. But because the pathway and the project are unrelated, they would only graduate with an MSc in Biomedical Science. About 95% of our students take this route here. So take the particular pathway and a related project. In terms of the full-time course, so it runs over 1 full year. In blue are the core modules. So we have the molecular biology. Within semester 1, they take the molecular genetics and genomics teaching. And in semester 2, we have the proteomics and transcriptomics. Alongside that runs the project design and development module, where in semester 1, we look at students preparing a grant application, which is based around the project that they've been assigned with their supervisor. And then in semester 2, undergoing a series of pass-fail lab inductions to get them up to speed, ready for their research project. And we can see the numbers here relate to the credits that each module is worth. So this is worth 30, this is worth 30, 60 and 30 respectively for each one, totaling the 180 credits of the masters. So all students take the modules in blue, and then students decide to take one of the following orange ones, and it can either be the cancer biology pathway with the cancer biology and therapeutics in semester 1, followed by contemporary applications in cancer biology and therapeutics in semester 2, or the clinical neuroscience, basic and clinical neuroscience in semester 1, and contemporary applications in neurobiology in semester 2, or the third option is the infection and immunity pathway with the infection and immunity module in semester 1 and the contemporary science of infection and immunity in semester 2. All these semester 1 modules comprise the core theory, which then builds on what happens in semester 2, which is more practical based. And these modules also follow in with the pass-fail lab skills developments here that follow then into the project. The projects typically start around February, move forward to the end of this module here if the student has been deemed to have developed the required skills, pass certain assessment points for practice, health and safety, etc. They then transition on to the project, which starts around end of March and moves through to July. So students get a decent amount of time to be able to work on their project with their supervisor. This can be lab-based project, so the ones you would typically expect to undertake, doing work in the lab with maybe cell culture, PCR, bacterial growth, etc. Or they can be non-lab-based projects, so a
You are breaking in between, Dr. Philip. Dr. Philip, we could not hear you. Dr. Philip? Dr. Philip, you hear us? Whether you hear us, Dr. Philip? We could not hear you, Dr. Philip.
Discipline that you've already gained previously. This is just a quote from one of our previous students. This is James, who was on the MSc a few years ago, and he's taken what he gained from the MSc, and he's actually working in the School of Engineering now with a project related to something that Matt is doing, which he might explain later on. In terms of teaching, so over the last few years, we've been taking some feedback from students in order to see what works best for them. A lot of our students either travel long distances, have children that they need to look after, or want to take on part-time work.
Again, your voice is breaking, Dr. Philip. Dr. Philip, we could not hear you.
Sorry, Dr. Deepak. I'm not sure if Philip can hear you, so perhaps he seems to be able to try and to reshare his screen.
Okay, maybe you can give your presentation, then we can also go ahead. Or in case if he wants to continue, I think he will continue now. Yeah.
Mm-hmm.
Yeah, let him continue. Your voice is breaking, Dr. Philip. Yeah. So we could not please go to the slide, no? Yeah. Yeah, now okay, but we could not hear your voice. Dr. Mathew?
Oh, yeah.
Dr. Mathew?
Hello.
Yeah, can you give your talk, like in case like if it is if he's facing some network issue? Yeah.
That might be a good idea. Phil,
Yeah.
Okay, we can't hear you. I can see your video. So what I'll do is I will now run through my slides.
Yes.
And perhaps,
that'll be a good choice. Yeah.
Okay. I'm really sorry about this. And perhaps if Philip,
no problem. Maybe he can give a. He has almost completed. Maybe he can give his talk at the end also. You can share your screen. Yeah.
Yeah.
You want to share your screen?
I'm not a host yet, I don't think.
No, I will make you as presenter now. Just a minute.
Okay.
Can you now hear me?
Yes, but there's a bit of a delay.
I don't know why that is. I'm looking at the network and I've got like 35 megabytes per second.
So what I'm going to do is now I'll go through my slides and I'll maybe come back to you at the end. Yeah.
Yeah,
Okay.
that will be a better idea. You are the presenter now, Dr. Mathew. Yeah, please go ahead.
Okay.
Maybe make it full screen. Yeah, that's perfect. Please go ahead.
Okay. Okay. Well, hopefully we managed to come back to you later, Phil, and hear more about the MSc program. But in the meantime, I'd also like to thank everybody for attending the talk today, and and for the invite to come and speak to you about some of the work that we're doing at the University of Plymouth. I'm going to talk about some of my research, which, as you mentioned, Dr. Deepak, is in antimicrobial resistance. So I'm going to talk about two particular projects, and these are quite well aligned with some spin-out companies that have come from the universities. So what I'd like to do is demonstrate that how we translate some of the research from the bench all the way through to ideally products that go into humans, and in this case animals, to try and address antimicrobial resistance. I'm not going to go into lots of detail, and I think there are microbiologists mostly in the audience, biologists, bioinformaticians. Hopefully I will cover in the next few slides things that are of interest to all of you. I'm going to dwell on this figure here. This comes from a report, it's the O'Neill review, which came out a couple of years ago in the UK. Perhaps you're familiar with this review. If you have an interest in antimicrobial resistance, antibiotic resistance, I strongly recommend the review. This is a figure from the review, and the headline from this review really was that if antimicrobial resistance is a major problem now, it's estimated, this is probably a low estimate, that 700,000 people die every single year because of infections that we can't treat with antibiotics. But in this review, it was suggested that if we don't manage to do anything about antimicrobial resistance, by 2050, 10 million people will die every single year because of drug-resistant infections, or infections we can't treat or prevent. And this will result in a number of other impacts on the global population and the global economy. So 300 million people would have had their lives shortened by antimicrobial resistance. This will lead to a loss of productivity, people not able to do their jobs because of either ill health or fatality. And it's estimated that this will take 3.5% of GDP, which is $100 trillion will have been taken off GDP by then. And the World have estimated that in this situation, 28.3 million people will be pushed into extreme poverty. And I guess clearly, obviously, at the moment, given the current pandemic, these figures may well be revised. And I think we're becoming very much more aware of the impact of infectious disease of all sorts on global health and our ability to deal with those infections. So given the time today, I'm not going to go into any more detail, and I hope this just emphasizes the fact of why we're looking at this as a particular area to carry out research in. Those are the striking figures for the problem that we face. But in the same review, the O'Neill review, there are a number of solutions listed. Jim O'Neill, the main author of the report, listed these 10 areas where we need to intervene in all 10 areas to be able to do something to reduce the impact of antimicrobial resistance. In the dotted circles, ellipses here, I'm showing the areas of research that at the University of Plymouth various researchers are involved in these different areas. The areas that I'll talk about today, though, are only in vaccines and alternatives and drug discovery. I'm just going to check at this point that you can still hear me okay?
Yeah.
You did? Okay, good. I just saw a hand raised just now, so I wondered if there was a problem. So sorry, the content of my slides today, I'm going to cover two case studies. The first one is vaccine development with a company called the Vaccine Group. So I'll give a very brief overview of the potential role of vaccines in reducing antimicrobial resistance. And I'll look at the approach we've taken in a major project to try and address antibiotic use in agriculture. I'll then close by looking at novel antibiotic development at a company that I formed 2 years ago called Amprologics. So I'll give an overview of the initial discovery of the main antibiotic we're developing at the company. Talk about the formation of the company, which has been required for us to be able to go through the safety evaluation needed to get into human trials. And then I'll talk at the end about some new discovery projects that we have running. And this will give me an opportunity to demonstrate some of the sorts of skills that project students in my group pick up while they're carrying out their research. So first, Dr. Michael Jarvis is an associate professor in the school, and in 2017 he formed the vaccine group. And the vaccine group is focused on developing vaccines for use in animals, and predominantly his interest is in combating infections that move from animals to humans, so zoonotic infections. And these include very severe infections like Lassa fever, and Ebola, hemorrhagic fevers. But Michael and I began to talk and decided that it would be good to apply some of his technology to address antimicrobial resistance. And the way we want to do that is by applying vaccines in agriculture, because up to 80% of antibiotics that are used globally are used in agriculture. So if we can reduce antibiotic use in agriculture, we argue we should be able to have an impact on AMR. And the inspiration for this for us came from a story that came from Norway. Norway farms salmon. It's a major industry in Norway, aquaculture, and generating farmed salmon is a key part of their economy. But if you're farming salmon in these sorts of pens in the open ocean, they will become infected with organisms like Vibrio. So to prevent those infections and treat infections, huge amounts of antibiotics were originally placed into the water, into these pens, so that the salmon could be protected from infection. But in the early and mid-1990s, a number of vaccines were introduced into agriculture in Norway. And the two resulting impacts of this, these purple bars here show the amount of veterinary medicinal products, basically the amount of antibiotics that were put into the sea to try and reduce infection in the fish. So with the introduction of 3 vaccines over the 1990s, you can see here the amount of those medicinal products, the amount of antibiotics have dramatically declined. And until today, the amount of antibiotics used is very, very small. But in combination with this, not only did the amount of antibiotic use go down, but the productivity went up. So the amount of fish being produced immensely increased. So this demonstrates that use of vaccines can reduce the amount of antibiotics without having a negative impact on productivity. So that was the inspiration for us. And we're working on a project that's been running for about 18 months now to tackle an organism called Streptococcus suis, which causes infections in pigs, in farmed pigs. And we're carrying out this project with sponsorship from the Department of Health and Social Care in the UK and the Ministry of Science and Technology in China. So we have 2 partners in the UK, myself at the University of Plymouth and Michael at the Vaccine Group. And we have 3 partners in India, Shanghai Veterinary Research Institute, Shanghai Yaotong University, and Pulaik Biological Engineering. And I won't dwell on the detail here, but across this group, we have expertise in vaccine design, immunity and infection with strep suis in pigs, and in manufacture of vaccines. So this group is now, we're working together to design a vaccine that is safe, inexpensive, and can be used as a single dose to stop strep suis infection in pigs that are being farmed in China. To do this, we're using a novel vaccine platform, and this is coming from Michael Jarvis from the Vaccine Group. It's a bovine herpes virus 4 vaccine vector. The bovine herpes virus is a benign, safe virus found in ruminant intestines. It's highly immunogenic. So if you insert genes into the virus and vaccinate an animal, they will mount a very strong immune response to the gene that you clone into the vector. That immune response includes both antibody response and T-cell response. And that's a long-lasting response, which allows us hopefully to have a single-use vaccine. And these are also very, very cheap vaccines to produce. To give you a little insight into the way we design those vectors and the way we choose the genes that we will include in the vaccine, we take the Streptococcus suis genome, and hopefully this will appeal to some of the bioinformaticians. It's a technology called reverse vaccinology. So we take the genome of Streptococcus suis, which has 2,500 genes. We feed it into a filtration system, a bioinformatics filtration system. Because we can't clone all 2,500 genes, we need to find a small number of candidates we can clone. First, look for the core genome, so genes that are present in every single strain that we can see of Strep suis. We then focus on genes that we know have products that are either on the cell surface or excreted from the bacterium. So that takes us down to maybe 200 candidates. We are focused on proteins that have a transmembrane region, because this is known to be good for inducing immunogenicity. And we have to have things that are pathogen specific. So we don't want to raise an immune response to something in the pig or to a different organism. And we then also don't want to have genes that mutate at a very high rate. So this brings us down from 2,500 genes all the way down to maybe 5 gene candidates that we want to clone those into the virus vector. And then we go through a number of assessment stages, looking at the immunogenicity and cell-based assays. We carry out challenge studies in pigs where we infect them with vaccinated pigs with Streptococcus suis. We then plan to scale up production and carry out field trials where we a full evaluation and then measure the impact on antibiotic use. So that's just an overview of that project, really. And it's quite a novel new approach to reverse vaccinology. But we hope within the 3-year project to have something that is ready to be commercialized at the end of the project. So that was the first example, and in the next example, I'll take you through the pathway from discovery of a new antibiotic compound to something that we are now commercializing and hoping to develop at a company called Antibiotics. I'll put this slide here to demonstrate the fact that in the 1940s and 50s, we used to be very good at finding antibiotics. Most antibiotics that we use now were originally found or are derived from things that were found in soils and other natural environments in the 1940s and 50s. And just this slide demonstrates that we were very efficient at doing that in the 1940s and 50s, but since then have become less good at finding new antibiotics. And that's led to a period over the last 30 years where we have essentially not found any new antibiotic classes to introduce to the clinic. So we urgently need now new antibiotics, and ideally they need to be from new classes so that current antibiotic resistance mechanisms don't compromise their use when they are used in the clinic. So this is an area, a real focus of my research and what I'd like to talk to you about now. And I'd like to talk about this compound called epidermicin NIL1. This is just one of the compounds that have been discovered in my group, and it's something we are very focused on developing for human use. Some of the data relating to the compound is shown here. I'll quickly summarize the data, but it's a very potent, highly potent antibiotic. It's probably 10 times more active at killing bacteria than most of the traditional antibiotics that we're now using. But although it's very potent, it has very low toxicity. So we can use very high doses without harming either human or animal hosts. we started to look at how this antibiotic epidermicin kills bacteria, and we know that it targets the membrane of the bacteria. And we just recently published the crystal structure of epidermicin and some work looking at the activity against membranes in this paper here. Before that, we published data showing that these are insect larvae, and we use these quite a lot in research in the lab. And this is the sort of thing that project students can be involved in, both at undergrad, MSc, and PhD level. These Galeria melanella larvae, we've shown that epidermicin protects them from infection with MRSA, a leading human pathogen. The most important data that's shown on this slide are the data on the right-hand side here. And I can't give too many details, but really all I need to say is that a single dose of epidermicin here was as effective as 6 doses of the current standard of care, which is called mupirocin. So this is unprecedented data. In this particular infection model, nobody has shown that a single dose of the compound can eradicate infecting organisms from the model. So this put us in a very good position to try and develop epidermicin as a product for use in humans. So the pathway that we've gone through to develop epidermicin from a very early observation on an agar plate of a bacterial colony producing something that killed the organisms around it. We've taken that all the way through development in the lab, development into use in an infection model, and now through to amprologics. To do that, we needed to submit patent applications. So there's expertise in the group for patent application and protecting intellectual property. We know how to assess what the best use for a novel antibiotic might be. We demonstrated excellent activity in the lab and in infection models. So this all justified forming a company to further develop epidermicin for use, which is what we're now doing. But just to kind of move ahead, really, we know that in the company we can't only have one compound, so we're now searching for new compounds, and we're doing this with marine biology colleagues at the university, Professor Kerry Howell. She sends these remotely operated submarines down to deep sea regions to recover these amazing sponges. These are animals that are growing on the seafloor over 1 kilometer down in the Atlantic. And Kerry sends out teams to recover some of these sponges so that we can analyze them. We like to analyze them because we think they hold highly novel bacteria, and that these bacteria will be producing new antibiotics that work in new ways. And other groups have shown that this is actually the case. So this is a new area of research for us, and one that we have project students at MSc and undergrad and PhD level working in. So this is what happens when Kerry visits a site in the deep ocean. The remotely operated vehicle recovers a sponge and harvests it into a collection tube. So the sponges are then returned or taken to the lab, where this is Matt. He's a PhD student with myself and Phil. He has a sponge here, and I'll now take you through the process of analysis that they carry out. So they homogenize the sponge material into a liquid, and they plate this out on agar plates. And for the microbiologists in the audience, I don't know how many of you have seen colonies that look quite like this. These are quite amazing bacteria that we see on these agar plates. This one actually appears to have a star shape on it. It's quite incredible. So some of these bacteria are very novel. We know that from the analysis that we've done. But one of the first things we do is look for an antimicrobial screen. So we plate these colonies of bacteria out on a lawn of bacteria and look for a zone of clearing. Anything like this that we're interested in, we then go ahead and sequence the genome of the organism. There are 2 routes to try and follow this interesting observation up. firstly, we sequence the genome, or also we can go through a process that I'll describe in the next slide. If we sequence the genome, we can use software tools to analyze the genome and identify gene clusters that we think are responsible for production of antimicrobial compounds. We did that with this particular genome, identified a gene of interest here, identified what the product of that gene was, had some of that synthesized, and dropped it onto a lawn of bacterium to show a zone of clearing demonstrating antimicrobial activity. Also, the microbiologists in the audience will probably be aware that if you take a sample like this, you plate it out on agar, you'll only grow a very, very small proportion of the bacteria that are actually present in that sample. So the other thing we do is use a genetic approach to directly sequence the DNA from these sponges, and again, take it through the software-based analysis to identify gene clusters. So we have culture-based studies and genetic in silico studies to try and identify genes of interest. The other thing that we can do with organisms like this, and this again is all the kind of work that students would be involved in, is carry out a purification-based approach. So if an organism like this is producing a compound we're interested in, we'll grow it up in a broth, and we'll into that broth, the organism produces lots and lots of compounds, and you can see that here. One of those compounds probably is the compound that we're interested in that's causing the antimicrobial activity. So we use a range of chromatography columns, some very simple like this, and others based on equipment that we have in the lab. These columns are held here on these pieces of equipment. And going through the steps of chromatography, what we do is essentially go from a very complex mixture to a more and more simple mixture, ultimately ending up with a single compound that is the compound of interest. And we then analyze that against databases to see whether we have a new compound or not. Matt and Poppy are both PhD students with myself and Philip, and they're both working in this area, and this just summarizes the work they do. The purification-based approach and the genome analysis and synthesis-based approach and characterization. So these areas in green, these are the areas that project students will become involved in. The downstream development, that's where things get more complicated and is carried out by other researchers. If you want to find out a little bit more about that work, I gave a TEDx talk in 2018 on finding new antibiotics in the deep ocean, which is a more of a kind of introductory level talk and maybe of interest to some of you. So with that, I'll just thank the people who have been involved in this work, our funders, and thank you for listening. Happy to take questions about specific points and perhaps come back and talk in more detail with anybody who is interested in specific areas. So thank you again for the opportunity. Perhaps now if I stop sharing my screen and hand back over. Have I stopped sharing my screen? There we go. Should I now hand back over to Philip?
Yeah, that will be good.
Can you hear me?
Yeah, perfect. We can hear you.
Okay. I don't know what went on there. Okay, sorry about that.
No worries.
So I can't share the screen at the moment. It's blanked out. It says I need to become a presenter.
No, Dr. Upton, Mathew can just make Dr. Philip as presenter. Can you do that? Just click Dr. Philip and then make him as presenter. Since you are a presenter right now, you have to change him.
There we go. Thank you.
Now it's done. Yeah.
Okay. Can you tell me whereabouts you got to before you
The same
couldn't hear me?
Typical timetable.
Okay, yeah. So I'll carry on from here. So apologies for that. It just happened to dip out. So as I was saying, we experimented with last year a compacted timetable where most of the teaching is on 2 days. So we can see here the core modules one of the pathways in green here and one of the other pathways in blue. When the third pathway starts, which is the clinical neuroscience, that will come alongside either green or the blue pathways. But what this essentially allows students to do is to prepare for work that we want them to bring into the sessions. It also allows them to be able to do other things. Several of our students do have part-time jobs, and they like to be able to know which days are available to keep that schedule. But around this Monday, Tuesday and Friday, while there is no timetabled sessions specifically, this is generally when students would be doing work on assessments, reading, or any preparation for these timetabled sessions. In terms of assessment for the Masters, so we have no formal examinations, so all of it is coursework based, but it's about trying to get students to demonstrate understanding of evidence, the comprehension of that data, and then taking that data forward through critical analysis to come up with some form of judgment or opinion that they can form from themselves, as well as what's in the literature. There are a number of different types of assessments. So some are timed assessments. So oral presentation, for example, might be a 20-minute presentation in front of their peers about a particular topic. The pathways do have things like an in-class test. So a certain amount of time is given for a student to be able to process the assessment. Some of the other assessments you will see here might be more familiar, things like essays, literature reviews. We do have case studies where data is given to the student and you're expected to form some kind of final opinion based on that data. We also have grant proposals. We try and emulate what we see in real life, so what academics would experience. So the project proposal forms the structure of that, similar to a grant proposal, to try and win some money. There are workshop exercises that also take data to do some kind of analysis of that. I mentioned before on the research, design and development core module that there were a number of pass-fail elements. So that is about attempting to develop the research, the understanding and the safety paperwork through to their project. And there's a number of pass-fail slots or gates that students pass through to demonstrate competency. And then students undertake a project and they would write up a project dissertation. This is very much like a paper that you would submit to a journal article and then submit finally to a viva, so a 30-minute oral defense of the work that they've been doing for the last few months on their project. Just a few of the student comments over the last few years. I've highlighted the bits I want to pluck out here, but it's mostly about the improved breadth of study that students get to experience as a result of this smaller group teaching. So they get topics available to them at masters that they might not experience at undergraduate level. They enjoy the good support that they get from their educational supervisors, and a lot of that is partly down to the small group teaching that we deliver. Because we're opening up on topics that are not necessarily available to undergraduates, students are finding this more engaging and more stimulating, and that's in part because of the specialist modules that we allow them to take through their pathways, but also the types of practical skills. We build on the stuff that students develop on their undergraduate course, and that allows us to undertake things that may be slightly more complicated and more labour intensive. So building up those skills both in the lab or potentially computer-based through bioinformatics. Our pathways are very much a reflection of the type of research we do. So Matt has already gone over some of the research that he does within his group, but looking at it from a broader perspective, our biomedical